Today's lab is about the cartilage tissue, starting with one type of cartilage, which is a hyaline cartilage. We will discuss the feature, the perichondrium, chondroblast, chondrocyte within the lacunae, the matrix and its territories, and these slides are stained by H&E. First of all, we have a low magnification for the piece of a hyaline cartilage. You can see the basophilic color of the cartilage. This is the matrix, and this is the site of the surrounding perichondrium. The perichondrium surrounding the matrix from all the sides, from this side and this side, and within we have the cartilage matrix with the cells. In high magnification, we can see the perichondrium, which is made by collagen fibers and fibroblasts. At the interjunction between the perichondrium and the matrix, we have a row of elliptical cells. These are the chondroblasts. The chondroblasts responsible for formation of new matrix. Within the matrix, we have lacunae occupied by other type of cells called the chondrocyte. So this is the perichondrium. These are at the border the chondroblasts, elliptical in shape, and within the matrix we have lacunae occupied by the chondrocyte. The matrix itself is divided into a basophilic structure that's immediately surrounding the lacunae called the territorial matrix, and the less stained basophilic cell color found in between called the interterritorial matrix. So we have the perichondrium, chondroblast at the periphery of the matrix, and within we have lacunae occupied by the chondrocyte, and the matrix divided into territorial and less basophilic interterritorial. The chondrocyte divided either single or in form of groups called isogenous groups. The growth of cartilage from within the matrix called interstitial growth from the chondrocyte group, and the growth from the periphery from the chondroblast called the oppositional growth. Then we have the elastic cartilage. We will discuss the same feature, the perichondrium, chondroblast, and the chondrocyte within the lacunae, and within the matrix we have elastic fiber, and stained by specific stain called the Verhoef hematoxylin that's specific for elastic fibers. Here we have a part of the external ear containing this piece of elastic cartilage. You can see the difference. Here the color is more dark, and we can see the perichondrium present on the periphery. In higher magnification, we can recognize the perichondrium, which is a dense connective tissue, and the cartilage matrix, which is more dark in color due to the presence of elastic fibers. At higher magnification, we can see the perichondrium and the chondroblasts at the periphery, and we have the matrix in which we have the lacunae occupied by the chondrocyte, and within the matrix we can see dark stained fibers of elastic fibers that is stained specifically by the Verhoef's hematoxylin. The last type of cartilage, which is a fibrocartilage, we have no perichondrium, we have no matrix, we have only dense collection of collagen fibers, and also we have chondrocyte within the lacunae that's stained by H&E. Here we have a picture of the fibrocartilage that's found within the intervertebral disc. We can see no perichondrium and no presence of matrix, only dense collection of collagen fibers that is stained eosinophilic in colors, and we have rows of cells that are the chondrocytes, so we have no chondroblast. In higher magnification, we can see the dense collection of collagen fibers that's found between the clusters of chondrocyte cells. These chondrocytes found in form of rows of cells separated by bands of connective tissue, which is mainly type 1 collagen fibers. This type of cartilage considered an intermediate stage between the hyaline cartilage and dense connective tissue, and found within the intervertebral disc. These slides are stained by H&E stain, and thank you.